Hello. Well, thanks to the handful of you who showed up. I showed up too, just. So your exam's this Saturday, right? Is it? No? 20. I thought it was the 12th. It's the 25th.
It's the last session. It's like the very last day of exams.
Oh, I'm screwed then. There's no way I'm going to have all of those marked in time for.
So yeah, I don't know how you're going to mark.
Yeah, I don't know either. Yeah, I'm totally screwed.
Just pass everyone.
Awesome. All right, well, today's going to be very short.
It's good for us because we have lots of time to study.
Great. So all I'm going to do today is go back over what's the format of the exam and look at a handful of example questions. So I've already gone over this in the prac class, so hopefully everyone's on board with it, but in case anyone didn't get that down, and so that there's a record of this recorded. Exam's 3 hours in length, it's 2 sections. There's 20 short answer questions, which you have to answer all of them. And you have to write 2 essays from a choice of 4 topics. And so 2/3 of the marks are from the short answer questions, so you should spend roughly 2 hours on those, which means about 6 minutes per question. And that leaves you about an hour for 2 essays, which means half an hour per essay. Bring along something to write with. Don't want to see you having to open a vein and scrawl it in blood. I've not yet marked an exam paper in blood, fortunately. Bring along a non-programmable calculator. There'll be at least, 2 or 3 questions that are calculation-like questions, sort of similar to the problem set. This is last year's exam paper as well in the library. In fact, I actually think the last 3 years' exam papers are available. So I would highly recommend that you look at those, because this year's questions are probably similar in style and format, and you may notice that some questions have a habit of reappearing occasionally in exams. So I think this is from a couple of questions, I think from the 2014 paper. Maybe it's the 2013 paper, I don't remember now. But this is, sort of the style of questions you get in the short answers, which is mostly, most of them are, regurgitate a factoid or 2 or 3. If you think about it, there's 20 short answer questions and 4 essays, and there's 26 lectures, which really is about 23, 24 lectures, because the first and last lectures don't really have anything. So if you think about that, that's roughly about 1 question per lecture in some way, shape or form. And so this first one, 5' and 3' ends of eukaryotic messenger RNAs are processed during and after transcription. In what ways are the ends of the messenger RNAs processed? So all I'm looking for there is for you to say that there's a 5' methylated cap and that there's a 3' poly-A tail. Tell me that. That's probably 4 marks. You don't need to write a half-page essay elaborating on the pathways of how that's done. I'm not asking for that. If you want to provide a little more information, that's probably not a bad idea. Question 2, DNA replication in E. coli occurs in a semi-discontinuous fashion. What does that mean? How does it occur? So, DNA replication, you've got to replicate both strands. DNA replication only proceeds in a 5' to 3' direction. I hope I've got that round the right way. And so you can just directly generate a copy of the leading strand, but the lagging strand, you have to synthesize in small fragments called Okazaki fragments, and then they're subsequently stitched together to make a complete copy with DNA ligase. So if you said all of that, that would give you 4 marks. And then there's usually a couple of questions where I want to, in addition to making you regurgitate random factoids, I want you to be able to think about how you apply molecular techniques to answer a particular question. So often they'll pose some little scenario and then ask some question as to what would you do, how would you do this. And so this is in this case, what is it? You've identified a bacterial gene, encodes an enzyme, blah, 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 blah. You want to see how it's regulated. So then you're looking for, in this case, this is a somewhat simple one, which it's just asking for techniques. Often I'll ask for an actual, I think more recently I've started asking for actual scenarios of how would you do this, how would you clone something, how would you do, how would you make a restriction up. In this case, it's just asking for what techniques can you give that could be used to study messenger RNA. And so you could propose doing QRT-PCR, northern blots, techniques to study whether the primaries, so S1 mapping and things like that. You could conceivably propose to use arrays or RNA-seq. You could make fusions with a marker gene to the promoter, things like that. Some other short answer questions. So what's chromatin? Why is chromatin remodeling important? So in that case, I'm looking for you to tell me that DNA is wrapped around around nucleosomes consisting of 5 histone proteins, and that these histone proteins basically cover up a large percentage, probably about 75% of the eukaryotic DNA, and so it covers important features like promoters, and so you need to have chromatin remodeling to move those nucleosomes out of the way so that the transcription machinery can access the promoters and transcribe genes. What else have we got? What are the AMP sites on a ribosome? Briefly describe their roles during the elongation step of translation. So you can tell me about what the AMP sites are, that they're where tRNAs bind, and you can describe how the amino acyl group gets transferred from one tRNA onto the growing peptide chain on the next tRNA. Question 8, maybe a little bit of a tricky one. The first part is easy, but the second part requires you to actually think a bit more, maybe. So, what type of damage does UV light cause? It causes pyrimidine dimers to form when you get covalent linkages between adjacent pyrimidine bases. And then E. coli has a photolyase that absorbs blue light and can directly reverse that change. In humans, humans and animals don't have a photolyase, so they have to use a different repair mechanism. The classic one they use is excision repair, where they'll cut out and it's nucleotide excision repair, where they cut out a region around the damaged bit and then resynthesize it. So, those short answer questions, I think, fall into 3 basic types. There'll be the, tell me, answer some relatively simple question about something covered in lectures. There'll be a couple that are, here's some scenario, what techniques, how would you go about doing what we're asking for in that scenario, and there'll be a few that are like problem set questions. The essay questions are different, and often they require you to synthesise stuff from across different lectures and bring it together to make a story. And so this question 1 here is a classic example of that, where you've got to remember stuff from probably about 4 or 5 different lectures and discuss how transcription differs between prokaryotes and eukaryotes. And then question 2 gives this long scenario, which really just asks how you would sequence and annotate a genome. So again, in this case, requiring you to remember stuff from both the genome sequencing lecture and the bioinformatics lecture, and partly aided by your what you've done in your Prac 4 bioinformatic project. And then this is only sort of an essay question, because really it's a series of short paragraph answers within it, pretending to be an essay. In this case, I'm just looking for you to tell me what are each of these techniques, what do they involve, and how are they applied. And so I don't want to walk through every one, but just as an example for southern hybridization, you describe how you digest DNA, run it on a gel, transfer it to a nitrocellulose filter, and then you probe that with a DNA probe for the gene you're looking for, and that's the probe will hybridize to homologous regions on the DNA on the filter, on the nylon filter or nitrocellulose filter, and then I'm looking for you to tell me what that's actually useful for. So in this case, you could say that this is useful. This has a number of applications, but it's typically for trying to find where your gene of interest is located. But it's also useful for DNA fingerprinting for forensics, things like that. Okay, that looks to be about that. Let me. I've put the recording back on. Those of you who aren't here, who knows what you've missed? But that's okay, because those who are... Statistics actually tells me that the vast majority of people don't even actually bother to listen to these recordings. Anyway, hope everyone's enjoyed the course. I guess I'll... Before I run out and leave you, if you've got any questions, I'll entertain questions.
The exam on the Sunday, the 25th, when
Yep.
was it held?
I have no clue.
So if you go onto iLearn, and there'll be a link for like exams, you put in your student number and it tells you where to go.
I got to the date and
Yeah.
the time of that thing.
That would have been the draft timetable, so at this kind of
Later stage, there's
at a
probably a
later stage,
location.
then they'll release the location up. So that should be released.
They're releasing it yesterday afternoon.
Oh.
There you go.
I wonder why I thought it was the 12th.
I've looked at it, I don't
I don't know. Well, I have no input. They just set the timetable without, any input from me. And that's I don't turn up to the exam either, but I have to be on call in case someone's got problems, or in case I've totally screwed up the exam and there's something that's not clear. I have to be on call in case someone has questions. I've only been called out once, which was last year, and in which case the question was actually totally stupid and pointless.
Actually, I only had one guideline, which is for statistics, and basically there was a slight change in one of the questions at the very start, which meant everyone had to redo half an hour, 45 minutes of work.
It's
Yeah, that was a bit like, oh. Because it flowed on that every single calculation after that changed.
any other?
How long do you want us to make the essays?
As much as you can coherently write in
Like
in half an hour. So I think a page or a page and a half is plenty to cover everything you need to cover. If you think diagrams will help, I would definitely include diagrams. There's no downside to including diagrams. I think diagrams are almost always.
Will we have to do something like color restriction now? I know you're not allowed to say collection that very color pencils.
Do you need? So if you feel you need coloured pencils to do a restriction map, I would bring in coloured pencils.
Yeah, it makes them look better.
Okay. If there's nothing else, good luck. And I may cross your paths at some point in your future scientific or otherwise careers. This is the last lecture. This is Thursday, and we're done.